Greetings. This is the last part 4 of the Sponges and Coral's digital notebook. I'm starting on page 15 with part 4. And we're going to go through the 7 functions again. Some of them are going to be combined, like respiration, circulation, and excretion again, just because of the simplicity of the animal. There's not true organ systems. So there's going to be some videos to watch and these slides, so you may have to pause as needed. So the first box there on slide 15 is about movement and support. So it says describe the hydrostatic skeleton. So most of these cnidaria have a hydrostatic skeleton. And what that is layers of muscles and water that actually hold the shape of the cnidarian and allow it to move. The hydro part refers to the water. So we're going to see some worms also have hydrostatic skeleton, in which they have this water that helps to hold and support. Medusa that do move, if you're picturing a jellyfish, that would be a good idea. They move by jet propulsion. So take a second, pause, and watch this video, which is on page 15, right by movement and support. The next section is about feeding. So if you remember that gastrovascular cavity, that's that opening, that cavity in the middle of the cnidarian. And so the polyps, they have their tentacles on the top, and the mouth is in between the tentacles, so it'll pull food down that way. And then the medusas are kind of flipped over, so you've got the tentacles on the bottom and the mouth in between those, so it'll pull food through that. There is a one opening, so this is food entering and leaving through the same opening. And in that gastrovascular cavity, there are some enzymes and such that will start to digest whatever is eaten, and then that digested material is what is taken into the cells. Respiration, circulation, and excretion. Nutrients are transported through diffusion. So once that gastrovascular cavity has ended up breaking down the food, it's diffused into the cells within the lining of the gastrovascular cavity. That's also how respiration and excretion occurs. And then one opening, so anything that's a waste will either diffuse like carbon dioxide through body walls or empty through that opening. Response, so there is, the sponges had really no nervous system. The cnidarians have a specialized nerve net. So they gather information from specialized cells, cysts, you remember that, statocysts. They have this nerve net with statocysts that are a group of sensory cells that help determine the direction of gravity. So that will help whether it's a polyp and its tentacles are facing up, or whether it's a medusa and the tentacles are facing down. Ocelli are eye spots, and they're just a collection of specialized cells, and they detect light. They don't necessarily see, like we see colors and shapes and 3 dimensions. Ocelli just detect light. So if there's an organism that doesn't want to be in the light, they can actually move away, they can respond. And then reproduction, there's both asexual and sexual reproduction. Some polyps are able to bud off, and you're going to see an example of that in lab. And that budding is just basically another branch that comes off, and it's a clone of that cnidarian. External fertilization is where the fertilization process of sperm fertilizing egg takes place outside of the body. And so sexual reproduction with some cnidarians is external. The last part, before we get into that, this is just kind of a really generalized life cycle of a specific kind of cnidarian called a hydrozoan. And hydra, they are, as adults, they're polyps, so they're sessile. So you can see that they can bud here, Asexually, it's just a branch that breaks off. The medusa is the larval stage in this particular animal, and that is the motile stage. That's actually able to move. Sexual reproduction can occur when the medusa stage releases egg and sperm. It's fertilized outside of the body, so that's external fertilization, and then that zygote grows, 2 cells become 4 cells, become 8 cells, blah, blah, blah. And then it anchors and becomes this polyp again. So this is just one type of cnidarian. And we're going to see an example of the adult in lab. Okay, the next section is about the diversity and taxonomy of the cnidarians. So this looks very much like the Porifera video in that this is the same. This picture right here is the same. This idea that, the sponges have multicellularity and heterotrophy, but so does everything beyond that. If you remember studying cladograms in Unit 1. But what also started to evolve was this idea of symmetry and true tissue. So cnidaria, they have radial symmetry, okay? But they are also not asymmetrical. So the sponges were asymmetrical. They also have some true tissues. If you remember that that one slide with the polyp and the medusa on it, and it had those different colored layers. Those were true tissues. Okay, and then our next section is going to be on worms, and so that comes out here, and they have simple organs in bilateral symmetry. The 4 main classes of cnidarians, again, there's not just 4, but these are the 4 big ones. These are going to be what you put onto slide 17 at the bottom, and they include class Scyphozoa, class Hydrozoa, class Anthozoa, and class Cubozoa. So you're going to look up examples of those in a picture of each of those. The next 2, or I should say the last 2 slides are about coral reef life. And there are videos there that I'd like you to watch in order to answer these. Actually, no. 1 second, and then you can watch the videos. The worldwide distribution, that depends. So this is the top of slide 18, the worldwide distribution of corals. So where corals are in the world is dependent on the temperature. They need warm waters. The water depth, some corals are able to live very shallow, like the reefs, and then some can live deeper. And the amount of light intensity. So that's kind of how corals are distributed throughout the world. So any kind of outside environmental factor that affects temperature, water depth, or light intensity will affect coral reefs. And so that's where these videos come in, and you talk about the threats in slide 19. So take a second here and pause and look at those videos, fill out the rest of these slides, and then come back after you're done. I did want you to come back here just to, if you stick with me through 4 parts of these videos. This is slide 8, and this is the answers to the top picture. If you want to double check to make sure everything is labeled correctly, that'd be a good idea. And then here's the bottom of page 8, and make sure everything's labeled correctly. All right, well, that's the sponges and cnidaria. Have fun.